Hi, I'm Andrew Ghazi. I'm a postdoc in the lab of Curtis Huttenhauer at the Harvard Chan School of Public Health and the Broad Institute. In this presentation, I'll be going over HALA, which is a method for multi-omic association discovery. This work was initially conceived of and implemented by Ali Ronevard and Kathleen Susipto, and the paper was recently published in Bioinformatics as part of this year's ISMB Proceedings. So what is multi-omic association discovery? So just to clarify what I mean by that, in modern biological sciences, this circumstance crops up pretty frequently. So the setup goes something like this. We first collect some high-dimensional data set X that has M features attributable to N biosamples. So these features are all the same type of measurements, so things like genotypes of SNPs, or metabolite levels. And then we collect a second high-dimensional data set, Y, that has MY features, but critically on the same N biosamples. And so the second data set is also high-dimensional, but it's a different type of features. So maybe it's microbial abundances or RNA expression or something. And we want to look for signals that correlate between the two data sets and groups of signals that correlate. So there are a lot of examples that you can probably think of from the literature. So things like methylation QTLs that affect gene expression, or eQTLs where individual SNPs are affecting expression, or things from microbiome sciences where the abundance of a particular microbe is associated with some downstream metabolite. And the question here is, the main methodological question is, how do we find these examples in the first place, given that we have such a large feature space in both of our two data sets? So kind of the simplest thing that one might start with is all versus all association. This is where you take all of the features in X, MX, and all of the features in Y, and you just make a big matrix of all of the pairs of these features. So for every pair, which is a cell in this large matrix, you compute the correlation strength and significance, for instance. So this has the advantage that individual associations are easy to interpret. So every cell of this big matrix is, you can kind of think of it as representing an individual scatter plot, like the examples I showed on the previous slide. And the downside of this is that given that both of our two input data sets are high dimensional and potentially have thousands of features, this matrix will be very large. So it can potentially have millions of cells, which adds computational burden and can really kill your power if you have low signal strength. And in biological circumstances, if you think about things like, for example, gene networks, many of the features in one or both of your data sets might be redundant. So they might be highly collinear. If, for example, it's the gene expression of many genes in a single pathway, they will all correlate very tightly. So an alternative approach is to use something like principal component association, where rather than looking at the raw features and comparing them, you first compute the top principal components for each of your two data sets. And you can think of this as like the dimension reduction is reducing the number of features you need to examine for associations. And so now when you compare the principal components of X against the principal components of Y, your feature by feature comparison matrix is now much smaller. So yeah, it isolates stronger non-redundant signals. And as I said, this matrix is much smaller now. The downside of this is that your associations that you get at the end, like the whatever hits you identify in this pairwise comparison, the associations that they represent are much more difficult to interpret, because now these aren't your original features, these are principal components. And so they're some complicated mixture of your original features. And so if you find anything interesting that you want to look at in your comparison matrix, you need to kind of backtrace back to your original features. And that sort of downside can also be said of other similar approaches that I'm not going to be talking about, things like CCA and Procrustes. They have a similar sort of downside. So Hala seeks a middle ground between these two approaches. It starts with the all versus all association matrix, but then uses the correlation structure in the two high-dimensional data sets to identify blocks of associations. So these are potentially groups of features in X that are associated with groups of features in Y. So what you get at the end is a representation of the entire all versus all association matrix, but now your features are grouped into these nice blocks that are more interpretable, which both aids in interpretation and deals with the redundancy problem and boosts your power for detecting true signals. So just to kind of go over how it works here, as I said, we start with our two high-dimensional data sets, X and Y. For this little cartoon, I'm only showing 3 features in X and 5 features in Y. But as I said, they could have thousands of features. And oops, I'm only showing a handful of samples, but again, you might have hundreds or even thousands. So the first step is to compute clusterings of the features in X and Y. And so this is with some similarity metric, like, for example, Spearman correlation. And then separately from that, we take the all versus all association matrix and find individual pairs of features that are FDR significant. Yeah, and then we use the hierarchies that we computed earlier, and then we use those to descend within the all versus all association matrix and find dense blocks of features. And so what we get at the end is a association matrix that has this nice blocking of groups of features. And that's what we report and visualize at the end. And I want to kind of dig into the step 3 here a bit more, just because that's kind of where all the magic happens. So to find the dense blocks of the associated features, we start with the all versus all association matrix, and we perform, we examine the associations in each pair of features and determine whether or not it's marginally FDR significant. And then we layer on the 2 hierarchies that we computed separately. And then we descend through the 2 trees. And if a block is dense, then we accept it. And if not, we descend through one of the 2 trees and then repeat the process iteratively. And so what dense means here is that at most a small fraction is not FDR significant. So at most, the small fraction has a blank cell. And this is what we call the false negative tolerance, or FNT. So when you're going through the descent process, you can, if you look at the whole matrix and it's not dense, like the whole matrix is here, you say, okay, we're going to split one of the trees, but we need to decide which tree to split. So here in this cartoon example, it's it's, very obvious that you want to split X, but the way we do this algorithmically within the data is to split based off of the decision that maximizes the change in Guinea impurity. So here, in this particular example, it's clearly choosing X will give you the biggest change in Guinea impurity. And then we rinse and repeat that process iteratively until we reach either a dense block of associations or singletons or empty blocks. And then these blocks that we get are what we report at the end. And so we've configured this all into a nice, easy to use software package. It's just, Hala, give it an X, give it a Y, and then tell it where to put the output. And there's a lot of user configurable parameters that can be changed to suit a given analysis. So things like transformation methods, discretization strategies, association metrics, things that you can, and there are things you can do to like speed up the analysis, or parallelization, or using p-value approximations. And it aims to be as general and as globally applicable as possible. So there's a lot you can do that I don't go into in this presentation. And it always tries to set common sense defaults that are sensitive to the structure of the input data. And just to kind of look at what the outputs look like a bit more here. Beyond just reporting and listing off the blocks and their statistics, we give some visualizations. So there's a plot that I've shown already a couple times. This is what we call a hologram. It kind of gives you a global overview of the blocks. And you can also set it to give diagnostic plots for each block. So if you want to dig into a particular block, it will show all of the pairwise feature comparisons for the constituent features of a given block. And it will do that for every block. It also, lists off the tabular summary statistics and things that you might imagine. So just to kind of get into the validation that we did a bit, we, of course, started with simulating data sets with the known block structure. This was done using randomized initial basis vectors. And this gives us features where we know what the blocks should be. And then we have these paired data sets where we know what the blocks should be. And then we hand those to Hala and see the positive and the true positives and false negatives that we get. And what we show in the paper is that Hala is more sensitive than Ala, so no H at the start. It's more sensitive, it more readily detects true associations. Meanwhile, it maintains the desired FDR levels. And as you adjust the false negative tolerance, you can tweak your sensitivity at the cost of a minor increase in false discovery rate. And we simulated across varying types of association structures. So data sets where the individual observations were just real valued and showed linear associations within the blocks, and more realistic data sets where we have mixtures of association structures along with categorical data and missingness. And we show that depending on your association metric and the association structure type, you can Hala almost always, or practically always, gets higher power and meanwhile maintains the desired false discovery rate. We also applied HALA to some real data sets just to as part of our other projects in the lab. So we applied HALA to data from the PRISM study, where it's comparing metagenomic abundances of many microbes against metabolite levels. And so this gives us a fairly large set of hypothesis blocks at the end to examine. But we were encouraged to see that among the top ranked results was a previously reported association between Ruminococcus navis and these fatty acid derivatives. And so it was good to see this result that recapitulated something we had already observed in a previous study. Right. And finally, I wanted to just kind of show some of the additional features of more exotic analyses you can do with HALA. So we came across this interesting example of nonlinear association discovery with HALA. This is in the TCGA breast cancer data set. So we found this by using the XICOR association metric, XICOR. And this association metric looks for associations that are based on any measurable function. So not just your simple linear associations. And then we looked at results that didn't overlap with the simple Pearson correlation results. And we found these three genes whose protein levels had this nice, seemingly parabolic relationship with the RNA expression of FOXC1. And so this was a bit puzzling at first because it's kind of a weird association structure to see that you might not expect at first. But then we noticed that when you overlay the subtype of cancer, that's explaining what this U-shaped relationship that we see in these three genes. And as far as we can tell, this finding is new. So the way you would interpret this is there's an increased FOXC1 RNA expression. The increased expression of FOXC1 seems to have a strong effect on the level of these three proteins in a way that varies with subtype. So in principle, one could have found this with mixed effects models, but that gives you a impossibly large model space to explore when you consider the number of combinations of RNA and protein features that you need to look at. Whereas with HALA, this was quite easy to find. Yeah, and so it's all packaged up nicely. It's part of the larger BioBakery suite of analysis packages. On the website, we have a nice tutorial and there's a support forum. Yeah. And so thanks to everyone in the Hut Lab, particularly Ali and Kathleen, who really drove the inception and implementation of this work at the start.